In this lecture you will learn about fluorescence microscopy, more precisely about microscopy in general, then basics about fluorescence microscopy, labeling, and the division of fluorescence microscopy techniques. We will take a look at a few examples of diffraction-limited fluorescence microscopy methods and at a few super-resolution imaging methods. So, microscope, the instrument to produce enlarged images of relatively small objects, can be based on different physical processes, like for example light, acoustic waves, X-rays, electron beam, and other processes. Enlarged image can be received directly, or digital methods can be used to obtain it. Important is to distinguish between magnification and resolution, where the former is the number of times the object appears enlarged, and the latter is the measure of the smallest detail of the object that can be observed. And for modern optical microscopes, the magnification can reach up to 2000 times with the resolution of 200 nanometers. Fluorescence microscopy, as it says in the name, is based on fluorescence of molecules. In most cases, laser beam excites molecules to higher states, and then electrons are relaxing to the ground state. They emit photons. This emission can be spontaneous or stimulated. And in fluorescence microscopy in general, this emission is collected, and the excitation light is blocked by using dichroic mirror, which directs the higher energy light and transmits lower energy light. Another important thing in fluorescent microscopy is labeling. To observe single biological molecules, a fluorescent molecule called label or a fluorophore can be attached to the molecule of interest. Then the most important thing is to choose the proper label. There are several important parameters which must be considered while choosing the label. First is photostability. For particular experiments, photoblinkable labels are chosen. In such molecules, after excitation, relaxation to non-emissive state occur, and then the molecule just relaxes to the ground state. Also, photobleachable labels can be chosen, which lose their fluorescence permanently due to the change in chemical structure due to interactions with the intense laser light. And these two types of labels are widely applied in super-resolution microscopy, as you will see later. And other parameters such like brightness, size, and so on needs to be considered too. And here you see some examples of labels for fluorescence microscopy. It's quantum dots, fluorescent proteins, and small organic dye molecules. And all of these possess a strong fluorescence and are used for different applications. The wave nature of light is fundamentally limited to the smallest object that can be observed using light microscopy. Limit was derived by Abe and states that any two objects can be resolved if the distance between them is approximately larger than half of the excitation wavelength. For example, considering numerical aperture of 1 and using green excitation light, two objects cannot be distinguished if they are closer than 250 nanometers. However, in last decades, many super-resolution techniques were developed to overcome the diffraction limit, and therefore fluorescent microscopy techniques are divided into diffraction-limited and super-resolution microscopy. And now I will present few diffraction-limited microscopy techniques. First is epi-illumination microscopy, which refers to any orthogonal scheme of illumination where excitation source is directed perpendicular to the plane of a sample. Illumination of a sample can be performed using many different light sources. However, strong divergence and incoherence of these sources allows effectively image only relatively thin samples. Other fluorescence microscopy technique is confocal microscopy. To improve thicker sample imaging in this technique, laser beam is focused onto a sample, and then a micrometer-sized pinhole for the emission path is passing only the fluorescence from the focal plane, while the light from out of focal plane is eliminated. To image one plane, a spot of interest is scanned across a sample. And for that, a few different techniques can be used, one of which is electronically controlled mirrors for laser beam, and the other is a spinning disc with multiple holes for deflecting a focus beam across a sample. And the third technique is total internal reflection fluorescence microscopy. This one is based on total internal reflection of light, and when the light is incident at the boundary of two media, it changes its direction of propagation. If incident angle reaches particular critical value, light does not enter into the second medium anymore, but undergoes a total internal reflection. And at the spot of this reflection, a evanescent field is created, which then propagates parallel to the plane of the boundary into the second medium. The distance for evanescent field to propagate is determined by the light incidence angle and refractive indices of the media. So in TIRF microscopy, this field is used to excite fluorophores which are close to the boundary, and the fluorophores which are further are not excited. Such a way of sample excitation gives excellent signal to noise ratio, and TIRF microscopy is widely applied in biology at a single molecule level. As diffraction-limited microscopy does not give access to nanoscopic molecular complexes, which can be of tens of nanometers in size, super-resolution microscopy can be a helpful tool to give access to the smallest objects. First, I will show one of the so-called localization techniques, photoactivated localization microscopy, which is called PALM. In PALM, weak short wavelength laser beam randomly activates inactive fluorophores, and then another longer wavelength higher intensity beam is used to excite these activated fluorophores, and the emission is then collected, and the positions of fluorophores is precisely located. The next step is to photobleach these localized fluorophores, and such a cycle is repeated: photoactivation, excitation, localization, and photobleaching. And then the image of localized fluorophores are overlapped with the other images until super resolution image is complete. Another localization technique is stochastic optical reconstruction microscopy, STORM, where the photobleaching of active fluorophores is eliminated for faster imaging. As in PALM, it is a time-consuming step. Actually, PALM and STORM share similar operation principles, like random activation and localization of emitting fluorophores. However, in STORM, photoswitchable fluorophores are used, not photobleachable as in PALM. Photoswitchable fluorophores can be then activated or deactivated, and the total super resolution image acquisition time is shortened. And now, a bit different from previous techniques, is stimulated emission by depletion microscopy. It is so-called spatially targeted technique and is based on the wavelength difference between spontaneous and stimulated emission of fluorophores. In STED, a laser beam excites a number of fluorophores, and then the second donut-shaped beam of longer wavelength centered to the excitation beam force majority of the fluorophores to go back to the ground state. Fluorophores in the middle of the second beam emit spontaneously, while affected fluorophores with the beam are depleted and emit stimulatingly. Both emissions have different wavelengths, wherefore they can be separated and emitting molecules can be localized. Then a scanning beam across the sample, a super resolution image is obtained. However, a drawback of this technique is the use of high laser powers, which requires special fluorophores which can withstand such high powers. And this microscopy technique was the first super resolution technique successfully tested on living animals. And another quite different technique from mentioned before is structured illumination microscopy, SIM. In SIM, sample is illuminated through periodically structured pattern, typically a grid-like high-frequency striped pattern. Then structured light interferes with the structures of a sample, and so-called moire patterns are created. Different images are formed by scanning and rotating these illumination grids, and then obtained interference patterns contain super resolution information about the sample we are studying. And the super resolution image is formed after non-trivial computational treatment of obtained interference patterns. And now I would like to summarize this lecture. We learned that fluorescence microscopy is a powerful tool to study minute objects, especially biological molecules and processes. And the sample not necessarily needs to fluoresce. We can use fluorescent labels for that. To image tens of nanometer sized objects, we can use a wide variety of super resolution techniques. However, for high spatial resolution, we might need way longer acquisition times, which sometimes goes beyond the timescale of biological processes. And also, high laser powers used for some techniques might not be compatible with the biological samples. The promising way to overcome these drawbacks seems to be a combination of fluorescence microscopy techniques, and here you see an example image of biological sample which was imaged with diffraction-limited TIRF microscopy, and then the combination of super resolution techniques, and we can see it gives us a valuable gain in resolution. So thank you for your attention.